going to move on now to our final plenary lecture, and I'd like to invite Professor Ineke Braakman to deliver our closing plenary, which is Unlocking the Key to CF, Understanding the CFTR Protein. Now, just before you start, we hadn't actually given some time for questions at the end, but please do post questions that you might have for Ineke in Slido, because we will give 5 minutes for questions at the end of your talk. Over to you.
Thank you. Thank you, CF Trust, for including me in this really marvelous meeting. I learned so much. It is wonderful to be part of this. And wow to the panel and everything I've heard here. I'm going to take you back to the protein. So this slide basically to show that I don't run the lab alone. I do this with my co-PI, Peter van der Sluis, and he's also my husband, which has some challenges, but most of the time it's fun. And so we are focusing on the CFTR protein. Now, this is a complex protein. It is large, 1,480 amino acids. It has 5 domains. You see here color-coded that they are 2 in the transmembrane domains. So they are in the membrane of the cell surface, but they start their life in the cradle of the cell, the endoplasmic reticulum, the ER for short. And there's a lot of domains, the blue and the purple, the nucleotide binding domains, in the cytosol. Now, maybe you're thinking now, okay, I've heard so many important things. Who cares about the protein? I'll find a bar, or maybe I'll go home early. I give you the opportunity to do so, but maybe you never stopped to think, or maybe you did stop to think at some point, how it is possible that modulators that were developed for only F508del or only G551D are actually working for over 170 different mutations, different variants. That's the protein, and I'll tell you precisely why. So, we know so much about this protein. This community has been working on CFTR for a very long time. This started with the discovery of the gene in 1989, and I'm really standing on the shoulders of giants, and I thank everyone in the room and outside of the room who did work on the CFTR protein and the cell biology and the molecular biology of CFTR, because we know what we know now because of the work of so many different labs. So because we know so much, and because we know it's a complex protein, I think a lot of people in the community think it is a special protein. But how special is it, really? It's not special at all. It's just like any other protein in the cell. And you would think, oh, bad luck. No, that's actually really good news, because we're not only standing on the shoulder of giants in the CF field, we're standing on the shoulders of giants in many fields. And this is just a short list of some of the major Nobel Prizes that were awarded for major processes, technologies like cryo-electron microscopy for useful for CFTR, on sugars added to proteins relevant for CFTR, transport through the cell relevant for CFTR. So all this and many more are totally important for cystic fibrosis and for CFTR. And so actually it's really good. We have a lot of extra knowledge without actually working on CFTR itself. And that helps to understand things. Now, this question of CFTR still is a challenge. It's a challenge to understand how the protein folds, how it gets its shape, but it's also a challenge for the cells. To place the challenge in perspective, basically every protein is made as a linear chain. So for CFTR, this is 1,480 amino acids, and it has to roll up, it has to fold into a functional machine. In this case, a regulatable chloride channel that also moves and works, and then still has to remain stable. So most proteins are at the limit of their stability because they have to be so flexible and so mobile. So these structures with AlphaFold and Rosetta Fold, another Nobel Prize from 2024, we now understand for so many proteins what they look like, but this was not derived from physicochemical principles. This was derived from language principles. So we still have no idea what the folding process is, and this is one of our major challenges. And a focus of our lab is to understand how proteins acquire and maintain their functional shapes from cradle to grave, because they're helped to get their functional shape, but then they have to also keep it until the end of life, and hopefully also a long life. Now, in a cell, this is a major challenge, because this wiggling about is a simulation, but it only tells you a fraction of the problems, because this proteins are wiggling about even at a higher density than this. And then it all has to be regulated, and proteins have to find their proper place in the cell. Now, a human body is around, as we are sitting here, around 7 to 8 kilograms of protein. But that's not one big clump, that's 10 to the 23rd protein molecules. Now, that's a mind-boggling number. All of them have to get this shape, but they actually constantly have to get their shape. So we have to eat protein because we need the building blocks, because all these ones in our cells, except for very few, are constantly turned over. They are degraded, they have to be made again, they have to find their shape again, and have to, everything starts again. So this is a challenge for the cell. It is that crowded. There's a lot of chance for clumping. And so what you see in a cell, this is a bit like the subway schedule, but then a bit simpler. But in reality, it's much more complex than even London tube system. Protein starts on the left, it ends up on the right, but it will also backtrack very easily. And what you see on the top and on the bottom are different fates that any protein can undergo. It can aggregate in many different ways, also toxic aggregation, and it can be degraded in many different ways. And actually, in a cell, without support, there would be very little chance of reaching the native state at the end. The other fates are much more likely. Now, fortunately, a cell has developed a very sophisticated system of what we call molecular chaperones, but there's many other support proteins that are surrounding the newly synthesized and the protein itself. Also, chaperones are proteins that are undergoing exactly the same processes as all the other proteins, and they are supporting the protein in each protein to keep their good shape, but they're also very nastily doing the triaging. They're deciding whether the protein reaches the native state or not, the functional state, or whether it's degraded or aggregated beforehand. So we all know this triage very well, because if you have the F508del mutant and many other mutations of CFTR, then it never reaches the final state, or it does so very shortly and then is degraded. So the triaging can also be a little bit excessively active, because if you rescue F508del, there is some functionality. So the cell has criteria to degrade a protein that are not necessarily coupled to function. Think, for instance, about the class 3 and 4 mutants that do reach the cell surface, even though they are not functional. Now, the good thing about folding 1480 amino acids is that the cell found a way to do this in a more secure way. And one reason is that the folding already starts during synthesis. And so what we found quite a while ago with the first PhD student working on cystic fibrosis in our lab was that the domains fold first. So instead of having to fold 1480 building blocks, you only have to assemble 5 building blocks, the 5 domains. Actually, 4 are really structured, and then R we tend to ignore for our studies because it warrants separate studies. The first domain that is being made is the transmembrane domain 1, and it's stitched into the membrane 6 times up and down, 3 times up and down, so you get 6 times strand in or helix in the membrane, and that's already folding as if you express it on its own. Then the next one to be made is the nucleotide binding domain 1, the the domain that is misfolded in the F508del mutation, and various other CFTR MBD1 mutations. And they already assemble, you see there, they get together at one point, they already assemble during synthesis. Now then the protein makes TMD2 and then MBD2, but it doesn't assemble until around 30 minutes after synthesis. So this assembly is not happening during synthesis, but what happens in between, or what actually happens, is that it's a two-phase process. Still, if you think about it, for such a complex, large protein that is part in the cytosol, part in the membrane, tiny bit of loops on the outside of the cell and in the endoplasmic reticulum when it's being made, this is a very simple folding process. On the left you see three domains, TMD1 and also the green TMD2 domain and MBD1. They fold already during synthesis, but they don't assemble except for that one little bit that I showed you before. They are folding as if you express them alone. So they have linkers that connect them, but it doesn't seem to matter so much. They really are fairly independent. And then MBD2 is still a bit more spaghetti. It really doesn't want to get any stability at that phase. But then, starting during synthesis, but for about 30 minutes on average after, the domains assemble. And then we see that TMD1 and TMD2 and also NBD2 actually really change conformation a bit. MBD2 gets stability, and then they become a well-assembled, in principle, functional channel. Now, this two-phase folding process makes it fairly robust. The question is, how do we know this? Well, we've used a tried and proven technique, which is radioactive labeling while the protein is being made. So we add 35S labeled radioactive amino acid building block to the protein. It then becomes radioactively labeled, and then we take the label away, and then you can just follow the protein while it's aging. So we take at a different age after the labeling, so in this case 0, 1, 2, and 4 hours of chase, we call this, but this is the age of the protein. We then lyse the cells, and we fish out from that whole cell soup with all these different molecules, we fish out the CFTR protein. And then you see that over time it changes. It goes from an ER band, which in the field is called the B band on the gel. It goes to the Golgi complex, and I call it here Golgi because it gets sugars modified, and then it ends up also on the cell surface, and it goes to other compartments in the cell. But basically the distinction is it's in the cradle, in the ER, where it folds. If it has assembled its domains, then it is allowed to leave the ER, and you get this also called C-band, the Golgi form. Now this is great. We can follow it over time when it's going through the cell, and but then we want to know, of course, what it looks like, because we found out that you first get domain folding, and then you get domain assembly. So what we do is use a technique that was pioneered in the field by Gergely Lukács and also Jack Reardon, but it was already basically from the 50s, limited proteolysis. Protein can be digested by a protease. Also in our gut, you just digest the protein and you get the building blocks. If you do limited proteolysis, low temperature, short time, little bit of protease, then you get fragments of the protein. So if you do this in a reproducible way, you get a reproducible fragment pattern. So what you see then, if there's more digestion, the protein is more loose and not folded yet, you get smaller fragments. If the protein is nicely packed and folded, you get larger fragments. And so in the gel, immediately after synthesis, we then fish out the fragments from the individual domains, so we can really look at what the protease did to the individual domains. And you see here, I blocked out the later ages. This is just immediately after synthesis. You see for TMD1, 3 fragments. For MBD1, there's 1 fragment, which is most of MBD1 actually. It's already pretty well folded immediately after synthesis. There's 1 fragment for TMD2, and there's a negligible quantity for nucleotide binding to domain 2. If you then look at the later times, you see you get larger fragments for TMD1 and TMD2 and for MBD2. So these three domains together, they assemble, they go at the same rate, they pack together, and they do so at the time the protein leaves the endoplasmic reticulum. We identified all these fragments. That was quite a major thing to do with the radioactivity, and we had to make many constructs, but people in the lab did a heroic effort, so we know precisely what every fragment is and where the protease in could hit CFTR and where it couldn't. And what we then identified is that during domain assembly, there were only 2 regions in the protein where there was a major change in the protease resistance, so the packing and the folding. And this was in the N-terminus, and for those of you who are familiar with the CFTR structure, the N-terminus is the first part that's being made, but that sort of embraces the protein when it's folded properly. So that process protects that N-terminus. And the other part is MBD2 plus the 2 parts of the TMDs that touch MBD2. So that part becomes folded and assembled. So these are basically small changes. Now, the next we wanted to do with that is just look at F508del and many other patient mutants. And by imaging, you can see that on the left, the wild-type protein is on the cell surface. So you see green outline of the cell. The red is basically a marker for the cradle of the protein, the endoplasmic reticulum. And you see some overlap because there's still some protein on its way out. But in the F508del case, you see there's complete overlap with the cradle. The protein just never leaves the ER and gets degraded. What it looks like in a gel is that you see on the left, instead of going to the outside of the ER and living outside of the cradle, outside of the endoplasmic reticulum, it gets degraded and you lose the protein. This is just because MBD1 is misfolded. Now, looking at the fragment pattern, I'm cheating now a little bit. I'm showing you the same gels, but now I'm blocking out the fragments that we are seeing in wild type, but we're not seeing in delta F. Basically, delta F mutant looks like it is just an early folding intermediate that did not assemble the domains. So MBD1 is misfolded, we don't get domain assembly, we get degradation instead of transport out of the ER. Now with that, we looked at the 60 most abundant missense mutants. And I won't show you the gels, but Marcel in the lab did an enormous effort to look at the fragment patterns of all of them. And it was almost binary. Most of them, or about half of them, looked a bit like wild type, which is probably because they're class 3 and 4 mutants. And all the other ones looked like they were actually showing no domain assembly, mutations in all over the protein, no domain assembly, degradation instead of export to the cell surface. Very strangely binary in a way. So the triage is fairly perfect. The protein, the CFTR mutant, leaves the ER anyhow. Either it assembles its domains and goes to the cell surface, or it doesn't assemble the domains and gets out to the cytosol and is degraded there. Now, one of the questions we had was, of course, what do the modulators do? They were developed, the correctors were developed for F508del, so the logical question was, is NBD1 rescued? NBD1 F508del is misfolded, it's degraded, we don't see a proteolytic fragment, so the question was, did it rescue NBD1 folding? And also low temperature rescued F508del at 27 degrees, you get the protein out to the cell surface. The answer is no. There's no rescue of NBD1 at all. Instead, there is rescue of domain assembly. The class 1 mutants do that by rescuing this N-terminus. Early on, before the embrace, they're basically stabilizing that N-terminus and stabilizing the packing, the folding of TMD1. So they're basically stabilizing TMD1, and they also work on TMD1 on its own. So by doing that, they improve domain assembly. But we all know the class 1 correctors on their own are not that impressive, so they need another one. And the other one is then, for instance, elexacaftor, which rescues domain assembly. So we see precisely what we see during domain assembly of the wild-type protein. So domain assembly is what's being rescued, not MBD1. So on the top you see with the corrector drugs, there's more domain assembly, there's more export from the endoplasmic reticulum to the cell surface, and so you get more function. And if you don't get more function, you need a potentiator. But at low temperature, there's a very different story because there's only a trickle of F508del leaving the endoplasmic reticulum at low temperature. But then the good thing is that little trickle makes it to the cell surface and then is stabilized by the low temperature. In part by the low temperature, but probably also by the total change of the cell at 27 degrees Celsius. But definitely the low temperature stabilizes instead of improving the folding for F508del. Now, thanks to structures and the work also of many other people, Ju Chen's structure is this one, where we know where these modulators bind. And they all bind in the transmembrane helices or at the membrane. So they're hydrophobic, they really like to be there. And the 661 is the tezacaftor, that's the class 1 corrector that improves the N-terminus and then also indirectly the embrace, and it also binds close to the N-terminus, so that makes total sense. But what we also saw is that ivacaftor, vertex 770, the potentiator, binds in a very similar way. So we also went out to see whether we could see a change in the conformation, in the shape of the protein by the proteolytic pattern. And indeed, we see that there is a big change, even 4 different changes that we see in the transmembrane domains because of ivacaftor. And for the correctors need to be added chronically to cells. The protein needs to improve its folding and then stability, and it needs to be there from early on. Potentiators in assays that colleagues that do functional studies are using is added acutely, and you see an acute change. You see this change also acutely. But 770, the ivacaftor, only works on the assembled protein, so we don't see any effect on the protein before it assembles its domains, but once it has assembled the domains, you can see with the ovals that I was drawing here and circles, the blue ones means there's protection, so more stability, more folding, more packing. The red ones are destabilizing. And so basically those 4 changes together mean that in TMD1 and 2 there's a bit of a shift, there's some sliding or so. There's definitely a conformational change, shape change that is fairly subtle. Now think about what I told you so far. F508del is rescued by the modulators, and many other mutations are rescued by the modulators because it rescues domain assembly. Domain assembly is compensating for defects in the domains. So that's why so many of the CFTR variants, the CFTR disease-causing mutants, are rescued by these modulators. If it would rescue only NBD1, then it wouldn't be such a good effect. So it's sort of a mixed blessing that the modulators don't rescue NBD1, which is probably why there's a potentiator needed, and hopefully future modulators will become drugs and will get into people with CF without having the need for 3 compounds. But so far, this is a lucky thing because it means many other people benefited from these modulators because they rescue domain assembly. Now, just to tell you then about what we know, and I can't even begin to say what we know about CFTR, so I just put a couple of things in there that I think are highlights of what we found, what we know as a field. One is that CFTR is a protein like others. General principles apply. if you are inspired by finding out what this is, go to the Nobel Prize website, look at all the different Nobel Prizes, and then they give you a tiny bit of text on what the significance was of that Nobel Prize, and basically a lot of it. When it comes to proteins, it always applies to CFTR. Second thing is that instead of having to fold 1,480 amino acids one by one, it goes during synthesis to a large extent and then is limited to a problem of having to assemble a number of domains. So first a phase of domain folding, then a phase of domain assembly. The reason it takes that long, around 30 minutes, to get the domains assembled is definitely because the cell has this whole triage and support system of molecular chaperones. We know that two heat shock protein 70 and 90, are absolutely crucial to get CFTR to fold. All of us that are healthy, don't have CF, basically would be suffering from CF and probably many other diseases if we wouldn't have these two chaperones. And even one of them already, if we lose a function of one of them, it's already enough to cause trouble. So they are binding to the protein while it is being folded, and they are delaying the folding process. So chaperones are actually delaying folding, but at the same time then improving the outcome of the folding process. The triage is binary. Protein goes out of the ER for sure, but it goes either to the Golgi, to the cell surface, and then is functional, or can be functional, or it's chucked out of the ER to get degraded. So the burden on the endoplasmic reticulum, on the cradle of the protein, is very similar in the healthy and the CF endoplasmic reticulum. Correctors and low temperature rescue domain assembly. They do not rescue F508del MBD1 folding. And ivacaftor changes CFTR structure. It's good to realize all modulators have a mode of action where they basically change the shape of the protein. They change the conformation of the protein, and thereby they change how the cell deals with that particular mutant, with that particular protein. Now, what do we still need to know about the protein? I mean, this is increasing understanding. I forgot to say I don't have any financial disclosures that are definitely not anything over the past 5 years. But we have collaborated with different companies because with our assays, we can actually tell them a bit about mode of action and also about whether different variants, different mutants respond or not. And one thing that we've had a paper not too long ago is to really look at non-responders. And I think that is a major question. What discriminates for every individual modulator, the non-responders from the responders? Now, in many cases, the assays are looking at function, either because a person with CF responds or not, or maybe a functional assay in organoids or in other systems. And what we then look at is whether the mutant responds biochemically in the fragment pattern. And the good news is that in most cases where we looked, we saw that they were responding to the modulators. For instance, the class 1 modulators improve twofold, and twofold a residual activity close to 0 is still 0. It means it's still twofold. So if there's another modulator that would crank up the residual activity, then that would still be a useful modulator to use for that particular variant, even though it's classified as a non-responder. The other thing is what explains the enormous variation in phenotypes per genotype. Now, this can be at so many different levels, all the Nobel Prizes, basically all the different processes that the protein undergoes in the cell, from synthesis, the translation rate, the synthesis rate, to the folding process, the help by the different chaperones. All cell lines, also the cell line that we are using laboratory cell lines because we have full control over them. Also the bronchial epithelial cells, they all have the same core system of supporting chaperones, co-chaperones, everything surrounding it. But of course, between all of us and everyone with CF, there is an enormous variation in all the expression levels, in the quantities of all these different proteins. That's what makes us unique, but it also means we have a unique makeup of all the supporting and triaging systems that these proteins need, that CFTR needs. So we need to understand better how they are changing the phenotype. And then what if genetic treatments are not enough? Maybe gene therapy will work, gene editing, one of the genetic therapies will work, but then we still need modulators, and then what modulators are we needing? And also with the read-through, the question that Margarida got is a very important one. For most mutants, it might be fine, it might suddenly be, upon read-through, a functional mutant, but maybe not. So we need to understand better what all these modulators are doing and what other therapies are doing. And then let's not forget, there's not that many papers, but there's definitely word out there that CFTR is not called a transmembrane conductance regulator for nothing. It really regulates many other channels, it binds other channels, and it also is known to bind several channels already in the ER and influencing their biosynthesis. So those are processes that we haven't even scratched the surface of, maybe totally irrelevant for people with CF, but I'm just holding off and thinking, let's not forget about that. So this is just a few of the questions that I think we should still answer. And I just want to end by thanking everyone who did the work. This is just a number of people on the left that all contributed to the basis of the conclusions I presented to you. Without the field, all these generous colleagues with wonderful reagents and the general support, or the generous support by the UKCF Trust to bring us together also with people in the UK and other countries, and the Dutch CF Society, Small Patient Society, Muco and Friends. We actually get twice a year, we get groups of patients and families and supporters to come into the lab for whatever they want, a demonstration, a talk, just a Q&A, just exchange of information. We learn from them a lot as well. And we are looking for postdocs and we're looking for a technician. So thank you so much for your attention and again for the invitation.